All right, so for those of you who didn't get to meet me yesterday, I'm Day2T2 Behbehani. I'm one of the postdocs in the lab. I'm also a hem-onc fellow in the clinical departments. And I'm going to talk to you today about some techniques that I developed to do cell cycle measurements by CyTOF and sort of the basics of how they work and why we need to use slightly different techniques to analyze cell cycle by CyTOF, and then some of the potential applications that you can use for these cell cycle techniques. So first I'm going to talk about the basic method for the cell cycle analysis, the markers that we use to mark S phase, G0, G1, G2, and M phase cells, some work that we've done to validate this with both cell lines and cycling T cells. And then I'll go in and talk about how we can use this to do sort of system-level analysis in normal and malignant hematopoiesis. People keep touching on the spade clustering, which no one's truly explained to you. I'll, again, touch on it without truly explaining it to you. Then I'll show you some data about normal bone marrow cell cycle states and compare that to some malignant samples. So why do we need to do something different for CyTOF? Well, this is the intercalator that we use in CyTOF, and from the labs yesterday, this is what we added at the very end. It's sort of an intercalating ring structure here that coordinates an iridium ion, and then this, in theory, intercalates into the DNA, and then we can detect the iridium. A couple problems with this are that it also non-specifically binds to proteins. So if you try and add the intercalator in the presence of the cell staining media, it doesn't bind at all. So it also sort of non-specifically binds to the proteins in the cells. The traditional DNA dyes probably do the same thing. It's just that their fluorescence is different when they're intercalated from when they aren't. And so the advantage with fluorescence is you only see the signal that's actually intercalated, because the spectral light absorbing and emitting properties of the things like DAPI and Host are different when they're intercalated as when they're not. But we don't have that advantage with the mass cytometer. And so as a result, if you compare the same cells by mass cytometry and this is Host dye by flow cytometry, you can see with the flow you get this sort of nice two peaks with the S phase cells in between that you're all used to seeing, but by CyTOF you just get a single peak. Now if you actually knew what phases of the cell cycle those cells were in, then you would be able to see that there actually is a difference. So if you compare the G0 to the G1 to the S to the G2 cells, it does change. Some of this is DNA content, some of this is size, but it's not enough that you can actually gate on any of that. So how did we get around it? So sort of a classical technology in regular flow cytometry is using things like BRDU, so halogenated pyrimidines. And this is iododeoxyuridine, very closely related, also used in the same way as BRDU and all the same assays you're used to seeing. In fact, it was developed about the same time, possibly even before BRDU. And a lot of the old antibodies will also bind to this, so they can be used interchangeably. So just a uridine molecule with the substitution of the iodine into the 5' position. And we can use this by CyTOF. And not only can we detect it by CyTOF, but we can directly detect it by CyTOF. Because the mass cytometer can measure the iodine, we don't need to denature the DNA. We don't need an antibody. Throw the cells in. As they get vaporized, the iodine signal comes out, and you can readily record it, even in incubations as short as 5 minutes. And as you'd expect, as you go to longer incubations, you don't really see much change in the S phase fraction, just an increase in the amount of labeling signal. So we typically will use 10 to 15-minute incubations routinely and can get really good data that way. So we had a great marker then for S phase. You can see here comparing the same cells, either with an antibody against IDU and fluorescence, and Host here, you get this sort of standard horseshoe shape, and that allows you to get G1, S, and G2. But when we do it by mass cytometry, we don't get that horseshoe again, because we don't have enough resolution with the iridium, but we can clearly pick out the S phase cells. We can actually do this with relatively low concentrations, though we usually work with the standard 10 micromolar that people use for BRDU and IDU experiments. So we needed some other markers. This is just sort of a summary of some of the important regulators of the cell cycle, and was a great sort of area to focus on in finding additional markers. So you can see as cells go from G0 to S phase, RB gets phosphorylated, cyclin D comes up, followed by cyclin E, A, cyclin B, and then M phase cells are also marked by phosphorylation of histone H3 during chromatin condensation. So a lot of these markers, in fact, all of these markers really are well established and used for flow cytometry. And so we wanted to see if we could use some of these to get good cell cycle resolution. So we tested several of them. So we tested cyclin B, and you can see this again is an iridium intercalator on the x-axis here and uridine incorporation on the y. And you can see that cyclin B gave us a pretty good resolution and really nice resolution relative to S-phase cells. For phosphorylated RB, what we really wanted to do is be able to tell G0 from G1. So one of the standard ways for doing this with regular flow cytometry is staining total RNA with like pyronin. So Hoesch pyronin is probably one of the more standard techniques. The problem is that regular RB phosphorylation sort of is generally considered a G1S checkpoint or response. However, there's recently published work saying that the phosphorylation at serines 807 and 811 correlated actually with G0, G1 transition. And so we investigated that, and we could get good resolution of that marker, and ultimately we were able to find that we could use it as that kind of marker. And then lastly, phosphorylated histone H3. We actually used the S28. The S10 is probably the more common, but this one actually probably works a little bit better. And you can see that that picks out the S-phase cells quite readily. And so from this, you can get a G0 population that's low in cyclin B, but not in S-phase. You can get an S-phase population by the IDU incorporation. And then you have these cells that are actually probably that are G2 phase cells that have high cyclin B that we can now discern from the G0 because we've removed the S-phase cells. And so that gives us G0, G1, S, and G2, sort of the standard horseshoe kind of plot. By using phospho-RB, we basically gate on any amount of phospho-RB that's less than the S-phase cells have because it comes on and stays high throughout the remainder of the cell cycle. And that can allow us to pick out the G0 cells. And then the M-phase cell gating is quite easy. And so this is a comparison of the same cells looked at both fluorescent and mass cytometry using the same markers and the traditional markers. And so you can see, again, we typically look at RB via IDU, and again, we gate on how much RB the S-phase cells have, and that allows us to find cells that have less than that, which are in G0. Then the cyclin B by IDU gives us a G1, S, G2 population. And if you actually look by fluorescence flow cytometry at the same cells, the traditional way with IDU by Hoesch, again, G1, S, G2. If you do pyronin by Hoesch, you can pick out this population that's really low in pyronin dye, which is your G0, and then all the rest of your cell cycle is here. But you can also add in cyclin B and phosphorylated RB, and again, you can pull out G0 cells and then do the same G1, S, G2 gating that you did by CyTOF. So we are really looking at the same things, and when you compare the percentages that you get by these two methods, you get really similar results. The CyTOF, since it doesn't have as good forward and side scatter, tends to see a little bit more debris and dead cells, and those end up looking like G0 cells, and so we do tend to slightly overestimate G0, and then subsequently, by percentage, underestimate everything else, but it's quite close. And so to validate this, we went into T cells, and then just took peripheral blood monocytes, stimulated them with PMA inomycin, and then took time points as the cells sort of got activated and entered cell cycle. You can see at the start, by both fluorescent methodologies, all the cells are at G0, so they're all in the G1, G0 gate of IDU by Hoesch, and they're all sort of low for pyronin Y. By mass cytometry, the same thing. In the cyclin B by IDU gate, they're all in sort of the G1, G0, and if you look at the S-phase cells, gate of RB versus IDU, they're all low. By 28 hours, you can see we start to get S-phase cells, again, by a typical Hoesch IDU incorporation. We start to see some S-phase cells, very few G2. Looking by pyronin Hoesch, we start to see lots of cells that have sort of entered the cycle, and then very similar results by CyTOF. And by 48 hours, we have sort of a complete cell cycle distribution with G1, S, G2 in both methodologies, estimate the number of cycling cells, though a large number of G0 cells persist, which is pretty common when you're doing PMA idomycin stimulation of T cells. And again, just sort of comparing the numbers, very similar distributions. So that gave us a lot of confidence that what we were measuring was accurate. So then what to do with this? Well, now, basically using 4 markers, of course we have other markers in these experiments that tend to confirm this, like cyclin A and Ki67, which I won't go into, but now we still have 34, 35 markers left to look at whatever else we want along with cell cycle. And that allows us to do really system-level analysis of really complex systems, either functionally complex when we're stimulating with damage, or immunophenotypically complex. How do you deal with all that data? I don't know if anyone's shown you this, but this is a slide that Aaron made. He'll probably show you when he talks. But these are all the possible plots you can get from a 4-parameter experiment. So biaxials are pretty easy to analyze your data. Go to 8. You can still probably do it with biaxials, but you got 28 plots to look at. Go to 14. Now you've got 91 possible plots. And when we're doing a 32-parameter experiment, it's pretty hard to just look at all the biaxial plots and interpret your data. And so that was what led us to sort of create or apply SPADE, or led Aaron, I should say. And the basic idea is that here in 3-dimensional space, again, Aaron's going to explain this in more detail, but if we have in 3-dimensional space this sort of distribution of a complex population, you can see certain areas that are dense, certain areas that are sparse. The first thing it does is it removes all the dense areas so that everything's sort of evenly distributed. Then it does a hierarchical clustering, and then it attaches those clusters in a minimum spanning tree so that the most related clusters are next to each other. The length of the line doesn't really mean anything here, just it's a question of which circles are closest to each other. And we also sort of size them roughly so the things that are more common are slightly bigger. And then you end up with sort of a tree structure. But of course, the computer algorithm doesn't know what a T cell is or a B cell. So what you ask it to do is say, of all these clustered regions, show me what the marker expression is on average in each of these clusters. And when you do that, you can see that it lights up very nicely. Like here we have CD3 cells, and then some of them are CD8, some are CD4, CD19 B cells, mature B cells within there that are 20. And so by doing that, you can basically manually annotate the entire tree. And so I did this with normal human bone marrow and was able to define 34 populations, I think. There's a couple that we couldn't say for sure what they were, but you can get a tree like this where we get like a T cell branch, B cell branch, monocyte branch, mature myeloid like neutrophils, granulocyte branch, and then some progenitor cells, including HSCs and early progenitors in the center. And this is just sort of what the individual markers look like, basically giving us the distribution we'd expect. And now we can look at those in detail. So here's the B cell lineage, and we can define sort of a pro-B cell that has 34 and some 10, but no 19, no 20. We can define pre-B1 lineage that has a lot of CD10, a little bit of 34, no 19, no 20. Then pre-B2, which is starting to get a little bit of 19, but still has CD10. And then sort of mature cells that have both 10, 19, and 20. And then we can look at the cell cycle and those cells at sort of a system-wide level. Here, these are sort of circle and circle plots. So the size of the inner circle, the red circle, indicates how many cells of that phase were in that population. We size it so that a completely full circle is about 67%, just so it's easier to view. And you can see that those cells are pretty much, the proliferating cells are pretty much concentrated in this pre-B2 population. Same kind of analysis can be done for erythropoiesis, and you can see that in the erythroblast population is where we're getting most of the S-phase cells. And looking at sort of the myelocytes again, you can see it's the early and late promyelocytes where we're getting almost all the proliferation. And then you can take this on to compare normal cells to cancerous cells and different cancerous cell lines. And so these are 2 AML samples. And when we look at a variety of markers here, I'm showing 34, you can see that the malignant cells light up quite readily. You can see here in this chart where I've sort of sized the circles to the absolute cell numbers so that the really rare populations are sort of invisible. But you can see that there's several different immunophenotypic populations in the leukemia, and some of them proliferate and some of them don't. which is interesting. Some of this had sort of been known before, but never looked at in this kind of detail. And a different AML sample has a very different pattern with different relative rates of proliferation. So very interesting technology from that aspect. And then we can also do sort of complex functional experiments. In this case, we're just using cell lines, so we don't really need surface markers, but that then allows us to look at all sorts of DNA damage response proteins, and then we can subdivide them by the cell cycle. And so you can see sort of here's what the total distribution of phospho-ATM, for instance, or phospho-H2AX looks like. But then when you subdivide it by cell cycle, you can see that the actual staining at different states of the cell cycle is quite different. And so this can provide you some really interesting ability to finally characterize these processes in multiple dimensions. So in conclusion, we've developed and validated a method for using mass cytometry to assess cell cycle state. And you can do it with only a few markers. The IDU is actually a marker you can't use for an antibody, so that doesn't even cost you a channel. So really, you only need cyclin B, phospho-RB, and phosphohistone H3. So only 3 of your 37, is it, that we're up to? Yeah, 3 of your 37 channels. So you've got still 34 parameters to look at whatever else you want in conjunction with cell cycle. And so with that, you can do sort of system-wide analyses of whatever process you're interested in. And then you can combine cell cycle measurements with other high-parameter experiments. And of course, just like to thank everybody in the lab who's been really helpful to me in getting this work done, as well as some of my clinical collaborators. And I'll take any questions. So Harris is going to talk about apoptosis next, and yes, he is using this technique to aid in his apoptosis, but of course you can combine apoptotic assessment with this information as well. What you do see, though, is as cells start to die, a lot of the markers just disappear. So when cells are really, like, have a ton of caspase 3, they basically all come out as G0 with this assay. I mean, if you did it by fluorescence, a lot of them come out as sub-G1 anyway, so it's not like, you know, it's hard to know what the cell cycle state was if something was dying. That's my hope, yeah. I didn't do PMA atomycin stems for those. Those are in vivo samples. So I went to the patients immediately after the bone marrow was drawn. I put it into a tube with IDU in it, incubated it for 30 minutes, and fixed it immediately. So that is how they were proliferating in the patient, or as close as we're going to be allowed by IRB to get to how they're proliferating in the patient. Any other questions?